Thanks everybody for joining again, for those of you I haven't talked to yet. So today we have a Zebrafish webinar, and we are lucky that two researchers have joined us to talk about their experiences and their studies. Christian Tudorache from Leiden University and Ruud van der Bos from Radboud University, amongst others. And also joining me today is my colleague Albert Willemsen. He will be co-hosting this meeting with me today, and he will introduce this meeting with a bit of information about zebrafish research tools. Some practical information. We have about 150 people that signed up for this, or more than that. We will be recording this session, so afterwards people will be able to view this again or view this if they're not able to join right now. We'll also have room for questions after each talk. Please use the question window to type in your questions, and then the Q&A window, and then we will try to answer as much of them as we can after each talk. Yeah, I think that's about it. So like I said, Albert will start with a short introduction. After that, Dr. Christian Tudorache will talk about biological clock functioning and personality types. And after that, Dr. Ruud van der Bos will go into sources of variation in zebrafish research and how to deal with them. So I hope you all enjoyed today. And Albert, let's take it away.
Right. Well, thank you very much, Gonnie, for introducing us and for organizing this webinar. I will make an attempt to share my screen. And then in the next 10 minutes or so, I'll talk you through the state of the art in zebrafish studies. Is my screen visible? No, not yet, I guess. And
there you go. Yes.
There we go. And now I will. Right. So I'll be telling you a bit about the state of the art, about tools and techniques for zebrafish studies. So we'll be covering a number of topics for different stages of development. To introduce myself, well, my name is Albert Willemsen, and I'm an account manager, and some of you may know me already. I'm originally a biologist, and but most of my working life, I've been in the ICT industry, and I've been 17 years with Noldus by now. And I serve a number of countries on this side of the Atlantic. For those of you who are not that familiar with Noldus yet, we've been in business for 30 years now. We're about 160 people. Headquarters is in the Netherlands. I'm in the Dutch office now, speaking to you. And we have a number of offices in other places. But what's much more important is that there's about 10,000 research labs working with our equipment and the countries where you will find people with Noldus equipment are colored green on this map. There's, it says here 98. I think we're now closer to, we're probably over 100 now. Some of our tools have become industry standards, especially the Atovision XT software, which is for video tracking, and it has about 2/3 of the publications on video tracking in behavioral neuroscience. Well, that's enough about the company. The first piece of tooling that I wanted to discuss with you is called DianoVision. Some of you may do larvae tracking, may do high throughput larvae screening, and that's this tool was developed for that some 8 years ago. It is also a video tracking tool. So you see here in this diagram, you see tracks of larvae in a well plate. I'll show you a little bit more of that. And here you see a well plate in a water basin, which is actually part of the DianoVision device. So you can track your larvae in this device. You see here in this chamber, you see a camera, which is used for the tracking. So it's a conditioned chamber. It's so there's the light conditions, temperature are under control. There's built-in stimulus life for light and for a tap. And that way you can standardize your circumstances and achieve a very high throughput. We put the camera very close to the well plate, as you see, and that is because then there is little movement in the device, which gives you a very nice and stable image. But to make that possible, there is a Fresnel lens. You see this here, this square item here is a Fresnel lens that is necessary to make that possible. I will not dwell too much on it, but the standardization and controlled conditions are important. Well, and here you will get an idea of what the results look like. You see now an image from the Daniel vision. We see the tracking, we see tracks building up over time, and you see already in a few seconds at face value that there is considerable amount of variation within this well plate, which may have to do with treatments or genotypes or whatever. By working in a 96 well plate, you could have 8 treatments and 12 replicates at 5 days post-fertilization. So you could do a whole toxicity experiment in one go, in one single trial. And that is a throughput, of course, that you could never achieve when you work with rodents. So that makes it very powerful and efficient. If we look at the results, well, I have an example here from the Erasmus University. It's an ethanol experiment, and the dark blue line is the activity of our control group. And we see a nice dose effect curve. So at at 2% ethanol, the magenta line, we see that the activity doubles, and then at 4% we see that it drops to half of that of the control group. But what's more is that we see that we've got 28 replicates, quite a lot. But look at these error bars. So at every point in this timeline of 15 minutes, where we analyzed in 1 minute time bins, we see a highly significant difference between the treatments. Already in the first minute, you have a high significance, and you also see that the within group variation is low, and the variation among the groups is high, which is exactly what we like. Taking it a step further, in a light-dark experiment, for instance, you see the same effect where the ethanol has a strong influence on the behavior of our fish. The blue line is the controls. So during the light period, the fish are much less active than during the dark because they prefer to be in the dark. So it's 10 minutes light on, light off. Light goes on again, we see that the fish freeze, but they also habituate quickly. And then at the next 10 minutes dark interval, they become more active again. Their activity drops when the light goes on again, etc. So we see that we see a strong effect of the light. We also see a strong habituation effect, and we see a strong effect of the ethanol. Again, we see it's all highly significant at almost at every point in time. And all of this in just 1 hour of tracking, and with 32 replicates. Temperature control is very important, and because our fish are exothermic, and so that means that all the processes are highly temperature dependent, all the metabolism, all the behavior, and that is why you need to keep the temperature under control. We do that by a water flow in this basin. So there is a laminar water flow, and there's a separate temperature control unit that supplies warm or cool water to keep the temperature within half a degree from what you specify. It's a separate unit, and that is because there is a pump in it that causes a little bit of vibration. And vibration is something that aquatic animals really hate. It scares them. So you want to avoid that, and so you can put this on a little side table apart from the device itself. But very important to keep it under control. And the temperature sensor here is inside the basin, so at the same spot where the fish are. You can also do video tracking on adult fish. And we see here an example of a memory experiment. It is a tea maze where the animal has to remember where there was a food item. On the right part of the screen, you see a heat map. So the fish has been spending its time on mostly in the right arm of the maze. And you can also get statistics for that, of course. And there's a large number of other tests that you could apply for cognition, for anxiety, for sociability, and many more. And here's an example for the social behavior. So many people do experiments on shoaling, where we use the inter-individual distances and approaches or withdrawals as a measure of group cohesion, and or social preference, where the fish has a choice between two groups of other fish or something else. These are all video tracking applications. If you work with, if you want to work with embryos or larvae and you want to do physiology experiments, that is also possible. And you will see here. So some people measure, for instance, tail flicks in embryos. Well, in this movie here, you see every once in a while you see a fish flicking its tail. Now, if you wanted to record that normally and manually, you'll be watching them one by one with a counter, and then say click, click, click when you see a flick. But you can only watch one fish at a time, which means that you would have to watch this movie 10 times. Well, we can automate it by measuring the pixel change, so the activity within the chorions, and this is fully automatic. We can also measure the heart rate in larvae. So you put them on their side in agar under a microscope. You see this pumping of the heart, and then we can apply a spectral analysis to the activity that we measure, and that returns us the heart rate. Well, there's more possible than just this, but it should just to give you an idea. I will give the floor to the scientists now. Should you have any questions, don't hesitate to send me a mail, albert@noldus.nl, and of course, you will find a lot more on our website. Thank you very much for your attention. Any questions?
Thanks, Albert. We can see there's one question coming in the chat box, actually. I would like to ask everybody to use the Q&A box as much as possible. She says, I'd like to ask about statistics that are optimal for these kinds of data. I use quite frequently the DaniVision, and usually I perform the test for each time bin. However, I'm not considering that in the following data, next time bin, the same embryos are going to be analyzed. So I think it's not the best statistic to use. Could you advise me on it? I don't know if there's a short answer right now. Otherwise, it might be good to get in touch with this person.
Yeah, maybe that is best that we do that separately, because there's a lot of statistics that you could calculate. It is usually more the question which is the right one than if E2Vision can do it. But
I will ask for an email.
Yes, please, let's do that offline. More questions?
I don't see any other questions coming in right now. Nope.
Well, then thank you very much. And then the floor is for Christian, I believe, right?
Yes.
Excellent. Fantastic. Very nice presentation, Albert. Thank you very much.
Thank you very much.
Yeah, I'm going to share my screen, as everybody does. And I hope this is what I want to show.
I can see it. This is perfect.
Excellent, fantastic. So then I just start. First of all, thank you very much for this great opportunity. I think it's very nice that Noldus organizes these seminars. I wish they were more frequent. This is the first one that I'm attending, actually. So very nice initiative. I would like to tell you today something about the biological clock and how different individuals cope with it. And this is basically a work that we published already a couple of years ago, but it is still very intriguing and still brought us more unanswered questions than we were able to answer. So this might be an interesting and exciting study where we use Noldus software. So coping with the clock. My name is Tudorache. I'm an assistant professor at Leiden University. And I'm especially intrigued by inter-individual variation in animals, meaning animal personality. And so, of course, first question that pops up when you tell these kind of stories during a cocktail party is, oh, but do have animals personality? And yes, they do. Animal personality is slightly different than our personality. And that is because in our personality, in human personality, that is the sum of values, the attitudes, the personal preferences, habits, political ideas, and so on. In animals, it's a bit different because we can't ask animals, what do you think about, for example, the recent elections in the United States. And the only way to find out what kind of personality type they represent is to subject them to behavioral tests, to behavioral experiments. In animals, we are talking about the consistent inter-individual differences in behavior and, what is very important, the underlying physiological setup. So it's a link, it's a correlation between behavioral traits and underlying physiological traits as a response to external stimuli. So how does this look? Let's say we have two behavioral traits, trait one, which could be risk-taking behavior or locomotion activity, and trait two, for example, aggression and boldness. And when we correlate these traits, each dot represents one individual, then ideally, of course, this is not a real data set. This is all beautifully made on PowerPoint in order to amuse you. In reality, this looks much worse. But when we find a correlation, then we can talk about a behavioral syndrome. Syndrome meaning walking together, where two behavioral traits are walking together. And this behavioral syndrome then again is representing the population, and the individual dots are in behavioral types, so personality types. And when a personality type is, for example, very risk-taking and very aggressive, then we are talking about a proactive personality type. And when the personality type is less risk-taking and less aggressive, then we are talking about a reactive personality type. Proactive individuals are bolder. They can thrive under constant predictable conditions, while reactive individuals, they are shyer, but they can thrive under unpredictable and changing conditions. So it is a trade-off within the population. It's not that one is the better one and the other one is the worse one. It depends really on the external situation. And what is very important as well is to see that these personality types are relatively different. So within a population, I don't know if my mouse is visible. Maybe somebody can give me a sign if there is something visible on the screen. Is my mouse visible, Albert? Can you see that?
I'm actually I don't see your screen.
Okay.
Even so.
You don't see my screen?
I see you, but maybe I'm doing something wrong.
Oh.
Oh no, I am doing something wrong. Yeah.
Okay. Excellent. But do you see my mouse now?
Yes, I see your
Oh,
mouse.
perfect.
Yes.
Fantastic. Then
Yes.
I just continue. So, for example, I always ask the audience, so what is this guy? Is he more proactive or reactive? And then some people say, yeah, it's kind of right of the middle, so he must be more proactive. And others say, yeah, but it's kind of in the middle, so it's really undecisive. The right answer is in relation to this guy, this individual is more proactive, and in relation to this guy, this individual is more reactive. So with other words, it is all very relative, and that means that within these studies, we always have to consider the entire population. And I will show you how that looks when we're actually doing our work. In any case, we are talking about individual variation, but what is important here is that there is a consistency over time and across situations. So we have to show consistency over time, and we have to show consistency over different tests. And this is actually what we are doing for all kinds of studies, looking at individual variation. Now, I'm using Zebrafish, the humble Zebrafish, and I want also to take this opportunity to hold up a beacon for the Zebrafish, which can be used in large numbers for preclinical trials, drug screening. The entire genome is mapped. We can study early development in embryos and larvae. It's a very robust organism, especially for stress research, what I'm also doing. But we never should forget that any model organism is a complementary model that never will replace one or the other. When they say, oh yeah, but rodents are mammals and therefore they are so much nearer to humans or monkeys. The point is that models are always working the best in combination with each other when they are used complementary. So with other words, one model never will answer the questions that we are asking, because by definition, as a model, it is not the real thing. Complementary models, and that is very important, are working when we are looking at the quality of that we want to study, and if this quality is represented in all models, and then we can see in how far this quality changes with their experimental setup. And so we can therefore assume that the same thing will happen in what we're modeling for, humans, for example. Therefore, these fish, they can be used as a reductionist approach to understand core mechanisms which are underlying, for example, a chosen phenotype, in this case, social behavior, and its fundamental features that are conserved over multiple species as a complementary model. So long story short, we use zebrafish. And what do we do with this zebrafish? How can we actually distinct different different personality types? So we have here a setup, and again, I try to go with the mouse. Yeah, exactly. So this is a tank, and this tank is divided by a little wall, and at the bottom of the wall there is a little hatch, and this hatch can open and close. Now we take a group of zebrafish, for example, 10 zebrafish that know each other. Mostly they are from the same batch, so they are brothers and sisters of each other. They grew up with each other. Here they are in these experiments. They are about a year or 2 years old. So they spend all this time together. They know each other very well, and their social interactions with each other, their rank in the group, and so on. So all these behavioral parameters are known to them, and they all come together. They are in this compartment. That is, when the hatch is closed, they get used to this compartment, familiar with it within about 10 minutes. You see that they behave totally normally. And then at some point, this hatch is opened, and one fish after the other emerges from this hatch. Now, the first ones to emerge are the ones that are bolder, that are more risk-taking, and therefore would explore an unknown and potentially dangerous novel environment quicker than individuals that are more reactive, more shy, and less risk-taking. And then when we take them out one by one, we have a rank, emergence rank, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And number 1 is the proactive one, the bold one. And number 10 is the reactive fish, which is less bold, which is more reactive, shy. And since we want to correlate behaviors, we also want to look at another type of behavior, in this case, aggressiveness. How aggressive is a fish? For this test, individual fish is placed in a tank. In this tank, there is a mirror. And then we are looking at approach time, approach frequency, and then also in the end, approach interaction, frequency of interaction. And then it is not only interactive behavior, but we are also looking especially at aggressive interaction as significant by, for example, biting, movements towards the mirror, rapid approach, and so on. And when we correlate all these ranks, so in this case, we have here 2 graphs. This is our emergence test in a group and alone, single emergence time. So how long does it take for this fish to emerge alone? And when we look at the link, the correlation between emergence rank, so in which rank they are when they're in a group, and the time that it takes them when they're alone, we see a positive correlation. So fish that are coming out later, else in an individual setup, need longer to come out in the same situation. And when we are looking then at emergence rank again, and this aggressive behavior frequency, so how aggressive an individual is, then we see that the early emerging fish are also more aggressive than the late emerging fish. So this is a very nice and convincing story for our point to make behavioral patterns consistent and therefore be able to talk about personality. Now, we were very intrigued by this behavior. I wanted to see what kind of genetic makeup these individuals have. So we took a couple of fish, proactive and reactive. We took their brains and we RNA sequenced their brains. They looked at the expression of RNA, the entire RNA in their brain tissue. And of course, almost 32,000 genes, which are mapped, came nicely out. Differences in proactive and reactive individuals. We saw that about 5% of these genes were consistently different, significantly different, which is actually not even that much, considering. But what was extremely exciting and intriguing was the fact that not the usual suspects would come out, genes that were coding for example, stress behavior or stress response or any other known aggressiveness and so on, but genes that were related to the biological clock. And not only that, so out of these 1,500 genes, there were about 3% related to the biological clock, which were mainly situated in the most differently expressed genes. So 20% of the top 100 differently expressed genes between proactive and reactive fish were genes related to the biological clock. And these genes I mapped here. So what we actually see here, there is a core loop. I'm going to this screen again. There's a core loop. This core loop consists of 4 types of genes: clock, BMAL, cry, and purr. And what happens is that these genes that are alternating, well, ticking effectively, alternating, expressing, they are expressing some proteins that are then again blocking the other set of genes. So clock and BMAL are producing these clock and BMAL proteins, and as a dimer, they are blocking the production of cry and purr proteins, which are then in turn blocking the clock and BMAL. And they are doing this rhythmically, so therefore we can talk about an internal clock. Now this core loop is then again stabilized by the so-called stabilizing loop, which consists of many different genes. And then on top, we have the light entrainment genes. They are genes that are light sensitive and are brought to expression by light. And so this is, well, the core clock and its stabilizing genes that are all related to the biological clock and its functioning. Now, the colors blue and red are genes that are coming to be expressed strongly in proactive individuals, the red genes, and in reactive individuals, the blue genes. And so, of course, very interesting. Now, the question was, what is the link between coping styles, between personality types, and the biological clock? We had three hypotheses how it was possible that these individuals or these different personality types had a different gene expression at the same time of the day. And these hypotheses were the link, so the link between the clock and the coping style. Is it that there is a phase shift? Is it that some individuals are living later than others? They are the latecomers, so to say. Or is it maybe that they have longer internal days and therefore there is a mismatch with the external clock? Or is it that basically there is a basic difference in the expression level of clock genes between personality types? Or finally, does it have to do with the fact that the amplitude differs between proactive and reactive fish? And of course, as you know, it's always the last one that's correct. So when we look at the expression of clock genes over time, and we see here again our nice clock with the clock genes, and then to the right, 5 different genes, 1 clock gene, 1 BMAL gene, 1 purr gene, 1 cry gene, and CYP-CA, which is belonging to the stabilizing loop. And we see very clearly that our proactive individuals, red, they have a very clear oscillation of the expression of clock genes in all cases in comparison to reactive individuals in blue. And when we look then also at the entire population, and this is something that I said before, it's very important to look at the entire population and to put everything in perspective, then we see that the area under the curve in the first row, that is the net mRNA expression, the product, so to say, how much RNA is there produced, and the bottom row, the amplitude of the expression, therefore the expression rhythmicity, are for all genes or almost all genes significantly negatively correlated with emergence rank, meaning that proactive individuals have a stronger, more RNA to produce of clock genes, so to say, and also a stronger rhythmicity in RNA expression. And of course, this was too intriguing to leave it there. So we were looking then at another level of biological function, and that was the production of melatonin and cortisol. Melatonin is a hormone that is responsible for the coordination of the internal clocks of different organs. In zebrafish, it has different functions, but that's another story. And cortisol is a very interesting hormone that, of course, for us, for stress researchers, but also for personality researchers, plays an important role because it is a stress hormone, but it also regulates a lot of other functions, metabolic functions. And what we see is again that our proactive individuals have a nice rhythmicity of endocrine function in melatonin and cortisol production. And again, when we are looking at the entire population, then we see very beautifully that the area under the curve is positively correlated. So more product, more cortisol, more melatonin is produced with later emergence ranks than with earlier emergence ranks, with later meaning reactive individuals than proactive individuals, and a negative correlation again with the amplitude with emergence rank, meaning that proactive individuals have a higher amplitude of the rhythmicity of this production, production of endocrine production of hormones than reactive individuals. And so, in order to make things round, we looked at behavior, and this is where Noldus Etovision software came in. We looked at how do individuals tick. We had a setup very similar as in the larval setup that Albert just mentioned now, but we rebuilt the whole thing for adult fish. So we had 18 tanks at the same time with camera on top with a red infrared light camera. And we observed these fish for, well, here 3 days, but the whole experiment lasted 6 days because we had another 3 days acclimation period to be on the safe side. Feeding was ad libitum, so there was no interaction, only night and day would change the light intensity. And we saw again that during night and day, our fish ticked much stronger when they were proactive than compared to reactive fish. What is interesting here is that our proactive fish, they seem to increase activity already when it's still dark. They anticipate the change of time, so they have a very strong internal clock, and they reach their peak in the morning, and then a second one just before going to bed, and they sleep well, and then again peak activity rises and they sleep at night. The reactive fish, however, they have these little peaks when light conditions change, so they are reacting passively, effectively, to the changes in light conditioning, and therefore to the changes between day and night. And when these conditions, quickly to say, we find the same patterns, again, a negative correlation in area under the curve, in amplitude, and this time also in rhythm strength with emergence rank. So rhythm strength, that is a measure for how repeatable a rhythm is over different days. So our reactive individuals, they have a weaker rhythm than our proactive individuals, and a lower area under the curve and a lower amplitude, therefore a lower rhythmicity. However, I also brought in this non-results, since they are very important. There is no difference in maximum activity, so that means that their capacity for activity is similar between proactive and reactive fish. It's not that the reactive fish are weaker and cannot move. And then finally, to make it a real clock, a real circadian experiment, we turned the light on dim consistently, so there was no difference between night and day. And then what we see, and this is maybe the most intriguing part of it, is that our proactive individuals, they still continue to tick well on the first day. On the second day, this ticking already goes down, and then the third day, they are really, really flat. And our reactive individuals, they lose this peak activity between night and day. So where this night and day, this change in lightening was, there is no reaction to it anymore. So without external stimuli, this is a pretty much a flat curve. And here again, in the circadian circumstances, there is no correlation anymore. There's still a bit of a correlation in the strength, strength of the rhythm, but it is much flatter. And this brings us to the conclusions. So what we can say is that correlation, there is a correlation between individual variation of risk-taking with the rhythmicity, the repeatability, and the variability on various levels of biological function. There is evidence for endogenous and exogenous control, meaning that our proactive individuals have a very strong endogenous rhythm, while our reactive individuals are depending on exogenous stimuli coming from outside, which is the change of lightning. And of course, from a biological, from evolutionary point of view, is there a difference in strategies? Is it good to have individuals within the same population that have a strong clock and individuals with a weaker clock at the same time? And this is, of course, very speculative, but we think yes, of course, as a biologist, everything what we find has an evolutionary, base. So yes, it is good to have individuals with a weaker clock that can follow the individuals with a stronger clock, because when changes occur for one or the other reason, then they are much more flexible and can react to these changes within the population. And of course, this work wouldn't have been possible without the help of my friends. And I would like to thank you for your attention.
Well, thank you so much. I really loved your talk.
Thank you.
We have, and we have a lot of other people saying that as well. So that's really nice to hear. We
Thanks.
also have 2 questions coming in. You can probably also view them yourself, but I will read them out loud anyway.
Look, if that's possible.
Yeah. So the first one.
Read them.
Sorry?
It's easier if you could read them, because I
Yes.
have the full screen mode, so I don't know.
Yes. I will read them out loud. Albert, I'll ask you if my connection sometimes drops. So if I'm being cut off, then maybe Albert can take over for me. So the first question is from Rafael Ayala Lara, if I pronounce it correctly. It says, thanks for your interesting talk. I was wondering if the emergence tests could be conducted in a larval and/or juvenile fish. However, if it requires a pre-established hierarchy, would the results be reliable?
Good question. We tried that. Actually, we built a little emergence test out of 2 Petri dishes, little 5 centimeter diameter Petri dishes that we connected with a tiny tube. And we had 8-day-old larvae that went from a darkened compartment to a lighter compartment. Now with larvae, there's another issue, because they actually don't like the dark. So it was more of a stress response, and how fast would they react to that. We saw correlation over time. So at day 6 to 7, and 7 to 8, we saw that these data were correlated, but they were not correlated with other parameters of stress responses. And so that was a bit of a dead end. So technically, yes, you can conduct these kind of things, but we had I think 1 paper published on that. It only worked in day 8. So the older they are, the more of a personality they develop. But still, we can talk about larvae when they are already externally feeding. So yes, in 8-day-old larvae, we saw that there was a good correlation with stress response.
Okay, so we have more questions coming in. The other one says, is from Anna-Katrien IJsselt. Absolutely fantastic talk.
Thank you.
I might have missed it, but how did you track proactive versus reactive fish in the same tank? Were they color-coded?
No, they were actually separate tanks. We had this little setup. We rebuilt the Danio Vision setup in big. And we had this camera on top with the infrared light, and then we had them separated in different tanks. So they were individually tracked.
Okay. Another question I see coming in is from Veronica. How do you measure melatonin in zebrafish larvae?
What we did was to snap freeze them, and then we would grind them up, and then we use ELISA. Well, we first is to extract melatonin because it is very little amounts available, and then we would use an ELISA to do that.
Okay, then I see one more question in the chat popping up from Saltuk. How many fish did you use while doing circadian analysis? Can you give information about the analysis using Etovision?
Oh boy, let's have a look. Yeah, well, you can have a look. So this is N is 72 for the behavioral parts. Here we have also 72 for the melatonin parts and the genes. Oh yeah, here also 72. So, well, sufficient, I guess. From a analysis, we didn't use the statistical function of Etovision. What we did was we extracted the data, and then we worked in other external programs, because Etovision is a great tracking software. Of course, I shouldn't say that too loud in a Noldus webinar, but they are specialized statistical programs that do a much better job in statistics. So,
and that's why we offer the option of export.
There you go.
Yes. So I think that's it for now. Let's thank you again.
Thank you
for this wonderful talk. And now we can hand over the microphone to Ruud.
Yeah, I have to get my presentation going. Thank you so much. Let's see whether this is going to work. Yes. I think there we are. Okay.
Yes.
Everyone can see my presentation?
I just saw it for a brief minute and now it's gone.
Not yet.
Oh, hang on, I see. I'll have to, there we go.
Switch the screen. Yes.
There we go.
There you go, yes.
You can also see my cursor moving on the screen.
Yes,
okay.
We can.
Thank you so much, Ronnie and Albert, for the invitation to share some of our results from the last years on the research that we've been doing. And actually this picture is taken from an outing that we had with all this, when we had this summer school that we organize each year. And again, thanks again for the invitation. Now, the structure of my presentation will be that first I'm going to say a few things about reproducibility. It will be sources of variation. Then I will be showing some of our data from the lab, and that will be on two different strains, AB versus Tupfer longfin. Both, I think, often used strains in research, and showing the differences and the similarities, and then how we have tried to deal with strain differences. Okay, I'll come to this picture, and I'm going to say a few words up front. Now, I have a degree in chemistry, and trained as a chemist, life is in one way actually that simple. Unless you are a very clumsy chemist in the laboratory, the outcome of a given synthesis is pretty much the same. And if there is any variation, it's often deducible to only a few factors, like pressure, temperature, or the like. Hence, we talk about controlled variation. So we have control on the variation of our data. So when I entered the field of behavioral research as a pharmacologist in the early 1980s, I noted actually a different story. It was time when people would breed their own animals, or even collected them from farms, like in cats, built their own equipment, like the Morris water maze, without paying too much attention actually to the conditions. Now, of course, inherent to behavioral research, there is variation. And that's not a problem in itself. Variation in traits is on which adaptation to the environment is actually built. That's what I think Christian showed. It becomes, however, problematic when there is uncontrolled variation. That is when we do not know the factors that contribute to variation in our data. That is the problem. So if there is a take-home message, I think, from this lecture, it would be that biological variation in itself is not a problem. It's uncontrolled variation. That is the problem. Now, I think the interesting thing is that over the years, I think that especially in the field of rodent research, much progress has been made in thinking about this problem. By using standardized breeding by suppliers, including well-defined inbred strains, standardized equipment, and observation techniques, such as I think Noldus has developed, like the phenotyper, as well as sophisticated statistics. I think, as was recently shown in an elegant paper on this topic by Kafkavi et al. In Neuroscience and Behavioral Reviews. So overall, this led to controlled variations. We identified the factors in rodent research which would contribute to variation in data. And they would come from the environment, they would come from genetics, or they would come from the interaction, or they would come actually from ourselves as being part of the experiment. So when I entered the field of zebrafish research some 5 to 10 years ago, I felt we were actually back to square one. Like as if all progress being made in rodent research had not entered this field. It was like the field was starting afresh without any knowledge actually coming from rodent research. And let me illustrate this with the following slide, with this slide actually. Now, one thing which was really striking, I think, there were no standardized test environments. So people would devise their own boxes, but didn't actually really look at the ambient lighting conditions, which would really contribute to variation, or the noise conditions, because zebrafish are sensitive to noise. And I see many people coming from rodent research moving into the zebrafish world, thinking about zebrafish as if they're rodents, but then swimming. But they're not. They're different animals, sensitive to different aspects of their environment. And it also means that thinking about what is a good test environment for an animal requires thinking about what is necessary for the animal to behave in a way which is natural to the animal. And there were actually no standardized equipment like we would have in the rodents. So the dynamic vision for larvae is one exception. But it would be very nice to have tests where you can actually bring the experiment to the animal, because now we're bringing the animal to the test. We select a zebrafish from the tank, and then we would bring it to the test by catching it and then releasing it into the new environment. Well, if there is one thing what zebrafish are very stressful to, that is being caught. And that we cannot habituate then to that, because as in rodents we can. Rodents in the end will habituate to being handled. But zebrafish simply don't habituate to that, which is a factor affecting our results. In addition, if you look at what people are doing, they have their own facilities where they breed their own animals. As said, that was done in the 80s with rodents, and we knew that variation was partly deducible to the fact that people had different husbandry conditions from early life to late life, with many different factors affecting that. So hence people then decided to use standardized conditions like the big suppliers have, and then actually purchase animals from that. But even when we would have animals in the lab, and we did some experiments ourselves, we still saw variation between labs, which were very difficult to track or to trace, because could have done with the lighting conditions, noise conditions. That's the same with zebrafish. I see many different systems being used that differ in ambient lighting conditions or noise or what have you, and we don't know the effects on that. There are no rules and regulations saying, well, this is the best condition, which makes a lot of difference, I think, when you breed your own animals. And then we don't have any really standardized strains. We have different strains. I will come to that later on, but they're not inbred strains. At best, they approach about 90% inbred. And that means if you start to breed from them and you have a difference in your environment, you're actually increasing the noise in your data. And if I look at the data that people publish, they often need quite a few animals to find significant differences. And that is because you have quite a lot of noise in your own data. So overall, if you look at the field where it is now, I think there is much to be improved by looking for a controlled variation. How can we actually, at the end of the day, have more grip on the factors that affect our results, and hence come to the situation that we have more controlled variation? Because variation in itself is not a problem, but if we don't know where the sources are, that is the problem, I think. So let me share you one data with this. This is coming from a study where they actually looked at Tupfalongfing. I will show you that later on, and wild Indian karyotype, which is another strain. And they looked actually at how much is shared between different labs and how much is different. So they're coming from the same supplier, if you wish, for TL and wild Indian karyotype. And you see there is quite a big difference between them. There is overlap, that's what they share, but there is also quite some difference. And that means if you start to breed from these animals, or if you have experiments going on, that doesn't necessarily mean that you will end up with the same results. I have noted over the times that experiments which seem to be that simple as zebrafish, we could not reproduce them. And then we noted that people either had different strains or their conditions were so different that it was very much difficult to reproduce those data. And I will come to that later on. And here's actually what you can see as a result of that. Now, if you look at the different strains or lines that are being used, well, the wild Indian karyotype is considered to be the most closest to the original zebrafish, or at least the wild type. Then we have the AB and Tübingen, which are both short fin, wild striped. But if you look at them at detail, they're really different. They're really different genetically. They're not interchangeable. We have looked at different experiments and you cannot simply replace one for the other. They're truly different strains, while they're both actually considered as being a wild type. There are two strains which are considered the best for inbred. That's about 90%, which is still very low if you think about that, but that's the best we can get. And then we have a strain which is often used, the Tupfalongfing. I'll come to that later on, because it easily reproduces. It produces a lot of offspring, but it's a mutated strain. There is a mutation, there are two mutations in that, and those mutations have an effect on their behavior and on their physiology, and that's what I'm going to show you. So now the story from our own lab, because we actually started with Tupfalongfing, because we had them in the lab. And I came into the lab at that moment that we had them. And even before I came there, it was easy, it was actually custom to purchase animals from a pet shop. Now, if there is any sin which you can do, I think, in behavioral research, is to go to a pet shop and just buy animals, because you have no control over the genetics, the microbiological status, or whatsoever. You haven't got a clue what you have in hands. But I still see people using pet shop animals in their papers, which to me is really, unless you may have a specific goal for that, but it's, at least in my view, coming from the background that I have, it's the worst thing you can do. There's because no control whatsoever about your starting material, which means it's very difficult for other people to reproduce your data because you haven't got a clue what the background is. So when we started with the Tupfalongfing, they actually came from a breeder, which I already was quite happy with. Now we looked initially at adult inhibitory avoidance learning. So the animals would be placed in this white box, and we would allow them to habituate for about 60 seconds. Then we would lift the slide door, and they would be allowed to swim into this dark chamber for about 180 seconds max. Now, most animals will move quite rapidly to this dark chamber because adult animals have a preference for this dark chamber. Oh, there we go. And if they would enter the dark chamber, this lid would be closed and they would receive a shock, a threefold shock. We use different. And then the next day they would be placed again in the white chamber. We would lift the sliding door and we would look at their behavior, which is a very simple rodent-based actually inhibitory avoidance task. Now, the thing that we set out, and I think that is what we prefer, to see which factors are actually affecting this inhibitory avoidance, because before you have a good test, you should know which factors contribute to the variation in your data. So we looked at different task variables. and we looked at different subject variables: age, strain, enrichment, and chronic stress, to be sure that the test that we had at hand would be really a test worthwhile for further exploring. And we would look at different readouts, behavior analysis, whole body cortisol, and gene expression in the telencephalon. So this is actually what we set out to have a really good grip on this task. And that, to me, is always one of the most important things. Once you start with an experiment, try to extract as much information beforehand just to see whether your test is actually working the way it should, so that you know that it's reproducible, that in the long run you have really something at hand. Now, we looked at two different strains. We had actually Tupfel longfin in our lab, and the Tupfel longfin has spots, and I will come to that later on, what the underlying genetic variation is, and it has long fins, in contrast to AB, which has stripes and short fins. And I will come to the mutations later on. And here we started with the experiment. So this is day one, where they are simply in the white box. We open the lid and we see how much time it takes, the latency, to move to the dark. And you see there is no difference. These are the individual data, and here you have the median. So there is no difference actually between the two strains. But then we looked at the next day, so when they received the shock, and you see that the Tupfel longfin, they have a strong avoidance response. So there is a strong increase in their latency, but it's not happening in the AB. They simply respond as if nothing happened. So we repeated another day. So they received the AB as both as the TL. Again, a second shock. So even those ones we forced actually to receive a second shock. And of course, the TL still had a strong response, but no response in the AB. So they didn't learn the trick. But why? Then we looked at a few other parameters. So we looked at behavior. So there was no apparent difference in the immediate response to the shock. But there was no 24-hour retention in shock learning. And there was also no habituation to the experimental setup, because we knew, or at least we saw, that when the TL would be in the white box, they would already behave in a different way. Whereas the AB would behave on the next day if nothing actually happened. So there was no retention on what happened the day before. When we looked at the physiology, the baseline cortisol levels were higher in the AB versus the TL. And there was no change in baseline cortisol as a result of the task. Also, when we looked at baseline expression of genes, there was an increased expression in the AB, and there was an increased expression also in the genes of the stress axis, and there was an increased baseline expression of genes involved in social interactions. So there were really two different strains. So then we decided to look further into this, because we felt like, okay, what is actually happening here? Why is this different? Now here we have the circle of life of the zebrafish. So starting from breeding, starting from the very first stages up until the adulthood, and then to mating again. So this is actually the circle of life of the zebrafish. And that's what we always work with, because we're interested in the different phases. And we decided to concentrate on the very first stages, because we felt that if there was already differences in cortisol levels in the adults, would it also then transfer to the offspring? Would we see actually the same differences already? Or do these differences emerge as the animals grow older? So we decided to look at larval behavior in more detail, and also larval physiology, and to really try to pinpoint the differences. Now, the interesting thing is what we found is that the cortisol levels in the IAB larvae were indeed higher than in the TL larvae. So they were actually the same as in the adults. So the AB larvae already had higher levels of cortisol compared to the Tupfel longfin. And also, I'm not going to walk you through all these different details of gene expression. If you're interested, you can look that up in the paper. But many genes differed between AB and TL, from the stress axis, from the immune system, from growth, and from all kinds of factors that affected development. So they're really different already at the larval stages. Then we thought, well, let's then try to see how their behavior changes, and let's take a very simple response: startle. which would be very easy to do in a standardized environment. And that's why we bought the Dino Vision system, where we would actually transfer the animals to the wells, I think as Albert already pointed out, and then transfer them to the system and look at their behavior. We thought of a protocol where they had 10 minutes of acclimation, 10 stimuli with a 20-second interval, leading to not much habituation in time, 10 minutes rest, and then 30 stimuli with a 1-second interval, leading to a very strong habituation. And here's just to show that we looked at maximum velocity as one of the parameters indicative of what we were interested in, that is the response to the stimulus. That's what you can see here. These are the 10 stimuli with a 20-second interval, and you see nicely the peaks as the stimulus is going on. So it's a very easy readout and very reproducible. But you also see here in this that the AB actually already habituated, and we'll come to that later on. Now we see the difference between the two strains. There is no difference in the first response. So they're both responding in the same way. But the AB habituates far more rapidly actually than the TL does. It's far more slower in its habituation. Very simple response, but a difference in the way they habituate. And we thought, why would this be the case? Then we thought, okay, let's make a proxy of this habituation. Let's divide this response by that one and take that as a proxy for habituation. So here we have that in a schematic way. So the AB and the TL. And then we thought, okay, TL has actually two different mutations. It has these spots and this long fin. Let's look at another strain which also has spots, which is actually a short fin. Well, in these stages, the short fin, a long fin doesn't really matter. But just to be sure, we looked actually for another spotted strain, and that's the leopard, which is spotted but has a short fin. And again, we see the same effect. They have a longer habituation, slower habituation. So apparently, this spotted pattern has more to do than just simply that it has it's not simply cosmetic, but it may also be another effect, and I'll come to that later on. And when we looked actually at the very first 10 stimuli, you see that the AB habituates very rapidly also to the 10 stimuli, whereas actually the AB and leopard don't. So we have a very robust difference between the two different strains, probably relating to the spotting. Let's look at what these spots actually, how they're being produced. Here we have the AB and the TL, and the spots are being produced by a mutation in a connexin. And connexins are actually providing intercellular contacts. So for instance, calcium is being transferred from one cell to the other by a connexin, and they're missing that. And that is why we have a spot on the skin rather than stripes. However, it's not only present in the skin, but it's also present in the brain, in the eye, and in the heart. So maybe we are affecting more than just simply the skin. So it's not only cosmetic, but it may also have a strong effect on the inside of the animal, maybe on its whole system. So that is one thing that we are thinking of, because if you look at habituation, and that is this slide, habituation is driven by NMDA receptors involving calcium transport in this part of the system. So here we have the starter circuitry, here we have the stimulus, here we have the motor neurons providing the response, and on the Mautner cell is actually where habituation occurs. And this habituation is dependent upon the NMDA receptor and calcium transport, involving, among others, intercellular transport. So then we actually looked at, is there a difference in the expression, or let's say in the activation of the NMDA receptor? So we used a few compounds which are sensitive for the NMDA receptor, and indeed we found differences between the AB and TL. Summarized in this statement, overall these data suggest that activation of the NMDA glycine receptor calcium channel complex involving these gap junctions is stronger in AB than TL, leading to more rapid habituation. So suggesting there is a fundamental difference in the way that they habituate, possibly due to differences in glutamate transmission and the way that it is actually being propagated. Now, this would suggest that TL may be more, well, they habituate slower, they're more sensitive to environmental stimuli, or they may be more anxious. Well, the interesting thing is that if you look at the novel tank test, which is measuring anxiety, and you look at strain differences, the leopard strain is indeed considered to be more anxious. It has a higher latency to move to the top. And it stays less, and it stays less time in the upper half as a consequence of that. And we know that this may be related to serotonin. So you see also here that using just a strain has an effect on actually what you're doing. So the leopard strain by itself may already be more anxious, as we also see in the tube for long fin. This long fin is also a mutation. It's a mutation in a gene which produces long fins, and it's actually just one of the mutations which can happen. Now, what does this mutation actually do? You saw in the previous slide that long fins also have an effect on the latency. Now, we looked at swimming behavior in a stagnant tank, so looked at their behavior for about 24 hours, then measured the distance they move per 5 minutes using the EtherVision system. And what you see here, this is the movement of the AB, and this is the movement of the tube for long fin during the day and during the night. And you see that per 5 minutes, either in non-enriched or enriched conditions, the TL simply move less than the AB. But what are they doing? We noticed that the TL was simply resting on their fins at the bottom. So if they didn't need to swim, they showed long resting bouts, just resting on their fins. Now you could ask why. Well, for one thing, long fins are dragging. So they're actually slowing your movement. It's more difficult to move with long fins. But also this mutation in the connexin, which is also present in the heart, where it might affect intercellular communication and thereby having an effect on the propagation of the heartbeats. So on the way that actually heartbeats are being made. So it could be that also has an effect on the heart. So if they don't need to move, they simply don't, because they don't want to spend energy. So that is one thing which, and we actually also put them in a tank where we had a current, and the AB would vigorously swim in the current, but the TL would simply drift. So they would simply drift into the water. They wouldn't swim in the current. So that means that they're not simply cosmetic changes, but they may have profound effects on the way they behave. And also, if you look at the social interactions, the social distance in AB is far more smaller than in TL. They are actually spreading far more apart. So also at the social behavior level, there may be differences between these strains. And I see people often using TL because they reproduce very easily, or they use them, well, interchangeably, or people actually make a combination of AB and tube for long fin, because then they have the advantage of reproducing quite a lot of the through the TL part, and then still have some of the wild type through the AB. And thereby actually you're enhancing the problem, because you're mixing two different strains which by themselves are already different, and you end up with something which may be a mix of whatever. Coming to the finishing part of my talk, because we were interested also in the early life stages, especially how cortisol would affect early life changes. And we looked at behavior, physiology, and immune system. We thought, well, what are we going to do? Are we going to use only AB? Or are we going to use AB and TL and look at what is common and what is different? So what is a robust finding common to both strains? And what is a difference? What is a finding which is only related to one of the strains? And how can we then learn what we can, how we can go from there? So we were interested in the question, what is the role of maternally deposited cortisol and GR in oocytes on the phenotype of the larvae? And we will look at variations in levels of cortisol, and how they would affect the balance in the different organ and physiological systems. So we would look at behavior, we would look at physiology, we would look at the immune function, and we would look at the way the heart actually functions. So we decided to use both TL and AB to deal with the problem of variation in these strains. Here we see the result when we have cortisol added in the early life stages and how that affects cortisol contents at day 5, so baseline cortisol levels. We see an increase in the AB, and that has also been reported by others. So if you add cortisol to the eggs only during 6 hours, in the very early life stages, so we expose the eggs to cortisol medium for 6 hours, then take it away, and then allow the animals to grow up in a normal way. Or other people would inject cortisol inside the eggs, and then thereby enhancing the contents of cortisol, and you will find an increased level of cortisol at day 5, which is also, if you measure that in the adult stages, actually remains. In the TL, that doesn't happen. And other people have used excessive amounts of cortisol to actually get it enhanced. So you see that the responsiveness also in this case is different. So would we have started with TL, we would have said, well, we don't see any effect. And you see again that the baseline levels of the cortisol in the TL is simply lower than in AB. So if you would have started with this one, we would have said, well, it's not going to work. And I also know from colleagues who have exposed eggs to alcohol, they were successful in AB to change the behavior in the long run, but they were unsuccessful in tubing it. And both are wild types. You couldn't see the difference from the outset. Well, you can, but then you have to look very specifically, because AB and tubingen look different, and they also behave differently. If you take time for observing them, if you have them in 2 tanks next to one another, you see the differences. But for most people, they think, well, they're interchangeable. So they use the tubingen or the AB. And as said, would my colleague have started with tubingen, he would have said alcohol exposure in the early life stages of zebrafish larvae doesn't have any effect on further development. But in the AB, it does. So there must be something in the tubingen why it's not working. And it's the same, I think, here in the TL. There is something why it's not working. There is something in the system why simply we can't see this effect. Now, if you look at the behavior, and again, this is the starter response, you see we find an increase in AB and an increase in TL. Both strains show a slowing of habituation. This is a common finding. So it's a robust finding. In both strains, we see a slowing of the response, which is not dependent on cortisol, on at least the acute levels of cortisol or the levels of cortisol which are circulating. Now you would say, if you would look at another test, because this could be indicative that enhanced levels of cortisol in the early life stages produces more anxiety. So the animals are more sensitive to information from the environment and hence habituate less strongly. So if you would look at another parameter like thigmotaxis, which means wall hugging. Now, anxious animals show a higher tendency for wall hugging, and we looked at that. The interesting thing is that AB show more wall hugging, which is in actually supporting these data. So they show more wall hugging, they habituate less strongly. But the TL, they show actually less wall hugging. So wall hugging is differently being regulated in these 2 different strains and is not in that sense a reliable parameter. So that's why we're actually looking at different parameters in different strains to see what is common and what is different. Now we also look at the immune system, and this is actually the only thing that you have to see from this picture. This is these are 4 different genes. Oh, hang on, I have to get back. These are 2 different genes, or 4 different genes that we actually used in a component analysis and clustered them in components. Here we have cortisol. We also use dexamethasone instead of cortisol to activate the very early life stages. The only thing that you have to see from it is that both in AB both in AB and TL, we've seen upregulation of these genes, AB and TL. We've seen upregulation of the activity of these genes. So again, this is a robust finding, independent of the strain, adding cortisol in the very early life stages or stimulating the GR with dexamethasone leads to an increased activity of these genes. So that's a common finding. And then we actually decided to continue with the AB because we felt like, that makes life easier when we go further. So how we try to deal with this variation is by looking at more strains to see whether we actually can reproduce data. Because I've noticed also in the field of the immune function, we had an experiment which seemed that simple. And we tried to reproduce it, but we couldn't. And then we actually asked the authors, well, what strain did you use? And they said, well, we used the Tübingen. Then we bought the Tübingen, nothing happened. And again, we asked them, well, what kind of strain did you use? Yeah, then we used Tübingen and then we crossed them with the pet shop strain. Well, we don't have this pet shop strain, but we couldn't reproduce their data. And then I asked a colleague in Leiden, well, did you ever try this paradigm? He said, yeah, it's not working in our lab. I don't know why, it's simply not working. So we're spending a lot of energy and time trying to reproduce other data, and we're failing because people don't realize that the background is an important part of the story. And that's what we have learned from rodent research, but seem to forget in zebrafish research. It seems as we are trying to start all over again and making the same mistakes that we have actually eliminated from rodent research. And that is really bothering me because it really takes away a lot of time and energy before we get somewhere. So I think and as said, there has been quite a good number of papers on how to deal with this. And I would really appreciate, I think, that we can profit from those insights. And me coming from rodent research, for me, it's quite natural to do that. But I see many people moving into zebrafish research, but without having the background in rodent research. And then they're actually jumping on a train, which is anything but smoothly running. And that's, I think, what to me the most important thing is in terms of reproducibility, unless we are, I think, making progress in this, zebrafish research in the end may die because of this variation. And I think that would be a sad message in a way. And I think with that, I would like to end my presentation by acknowledging, well, that's actually, I think, what I just discussed. Unless we know the different factors contributing and really taking care of understanding how they contribute to our data, then we can really make progress. And I would like to acknowledge quite a few people who over the years, I think, have obtained all these data and work together to get this picture in the end going. And with that, I would like to thank you for your attention on this late Friday afternoon, at least for the people in Holland.
Well, thank you so much, Ruud. That was a really interesting talk. I was already familiar with your research, but it's really interesting to see how much effect the differences in strains can have and how much that influences your research and reproducibility of your results. So thanks so much. I really liked it. I have a few hiccups in my internet connection, so I might not be getting all the questions in. I'd like to ask all the attendees if they have questions to put them in the Q&A box now or in the chat. That's also fine. Albert, do you see anything coming in?
I don't see questions in the Q&A box, but I do have a question.
Awesome.
So thank you very much, Ruud, for pointing out once again how important it is to standardize, and also showing how much difference there can be between the strains. And I was wondering if there has been any attempt to actually phenotype all these different strains, or and to actually get a better understanding of the differences between these strains in a structured way. And if not, that would perhaps be something that the scientific community could pick up.
Yeah, I think there are a few things to say about one thing is what I feel is that tube for longfin, due to its mutations, and I think especially the one in the connection really has a strong effect. So the question is it actually a strain that you should use for research? Unless you have a very simple mechanism that you're interested in, say enzyme X leads to product Y, and you have but if you want to make mutations in the sense of genetic mutations, I think that tube for longfin is just not worthwhile doing it in that mutation, because it by itself is already affected. It has already mutations. And the interesting thing is that those mutations will never survive, because both, I think, have a negative effect, and in the wild, they would simply disappear. There are spontaneous mutations, but they simply disappear because their survival is very low, because both are hampering their behavior and actually their whole physiology. So in that sense, I think I would never make a mutation in the background of a tube for longfin. I know these colleagues did that in the beginning, and they had difficulties in some areas. For instance, in the visual field, people had mutations affecting the eye, and they couldn't get it working. But it's, I think, because this connection is also present in the eye. And these connections have a very strong effect on intercellular communication. That is trafficking between cells, like calcium transport and what have you. And they're all very important for important aspects of heart functioning, brain functioning, and eye functioning. Now, if you make a mutation in that background, you're lost. In addition, I think the problematic thing at this stage, if you make a mutation in general in a background, even in AB, you have a variable background. So it's very difficult to pinpoint how your mutation is interacting with the variable background of your AB strain or your tubing, which means you may have quite a lot of noise in your data. In addition, both AB and tubing are actually missing part of the chromosome compared to original zebrafish, because in cleaning up a lot of these mutations, they chopped off parts of the chromosome, determining, for instance, whether they really become males or females. Now we have males and females, but it is because the factors in the environment determine much of whether they become males or females, but they have XY chromosomes, a form of XY chromosomes. That is what the original zebrafish has, but they're being chopped off that part in the AB and tubing. So AB and tubing become males and females far more importantly by the environment than actually by their own genes. So that means we actually have chopped off bits and pieces, and we haven't got any control on that. And that to me is still something which I feel we should address in one way or the other, or should think about strategies to incorporate that. Because without that, you're actually lost. You're wandering in the woods without having any compass where to go.
Thank you.
I see there are some other questions coming in. A technical question: how did you build the avoidance cage, the shocker?
Well, we actually did it in a very simple way. When we started, we thought, well, we can actually devise it ourselves, and we just took it from. We just built it from I think a kind of PVC that we had. But I have to look at for the details. If someone is interested in that, I can give you all the details of that. But it worked in our hands. But again, here, and that has been published. If you don't have the right ambient conditions, that is lighting conditions, you will simply don't get a really decent response. The contrast between the light and the dark box should be really strong, because if it's not strong enough, the animals don't show really reliable behavior. So that really means that you have to have a very strong contrast. And the problem with this task is that you have to handle the animals. And that was something that we felt like in the long run wasn't really productive. So we thought, can we actually devise a kind of home tank variation where we actually train the animals in the home tank. So we bring the test to the animal. And that's what I think always also already discussed with Noldus. And my view would be one step ahead in zebrafish research would be to get rid of the handling procedures by bringing the test to the animal, by seeing how we can actually devise home cage testing. And that's something that you can do, even I think with a inhibitory avoidance, because you can actually bring the test plates to the animal. And that's, I think, far more, in the long run, giving you far more stable results than bringing the animal to the test. But if anyone needs to have the details, I'm happy to give them.
Okay, thank you. We have a couple more questions, but we are running quite late already. Maybe, I don't think there's a short answer for this, but somebody asks if if you could recommend a strain for neurobehavioral studies.
I don't think there is. I mean, all strains have their own pros and cons. And the typical longfin, I think, is, because of its mutation already that it has, would be, I think, the least preferable. Because you have changed something in the internal part.
I think you froze
so
there for a
AB
second.
Tübingen would be. Sorry?
Yeah, you froze for a short second.
Yeah, so AB and Tübingen would, I think, be. But what I always would do is I would use both strains to be sure that your effects are robust. Because if they're dependent on one strain, you have a problem in the interpretation of your data. So I would use AB and Tübingen and then see if the results are robust or whether they're really dependent on one of the strains.
Yeah.
That will give you more information, I think.
I think that partly also answers another question, that is, would you advise against using the longfin strain for behavioral studies such as in anxiety?
Yeah, because I think it's both mutations will have an effect on whatever parameter you're measuring. Even the longfin by itself will have an effect on any behavioral measure that you're doing. So to me, the typical longfin is too much mutated to be really, I think, of interest, unless you're interested in the mutation itself. Because this connection mutation is by itself interesting, but then you should focus on that specific part.
Cool. Okay, let's do one final question. That's from Bavan Amin. It says, "Hi Ruud, very nice talk. I was wondering whether there really is a best condition. Don't you think too much standardization would make findings too specific, thereby making it difficult to extrapolate findings? What would you say the balance would be?" Thanks again.
I think you should always look for standardization that is, for me, standardization means controlled variation. It's not a problem that you have variation. It's if you know the source of the variation, then you can use that to your advantage. That's what has been done in rodent research. Some people would say, well, we can use different kinds of backgrounds, but if we know the effect of the different backgrounds, for instance, in the housing conditions, we use that as a factor which is controlled. So the point is not that you should have standardization eliminating everything, but the essence of controlled variation is that you know the sources of variation. And those factors you use actually to your advantage. And you can do that a priori at the start of your experiment. So look at different housing conditions and the effect on your data. And thereby you have a full exploration of what it actually means. Or in a meta-analysis, if you have many different experiments in many different conditions, you can try to do with statistical analysis, parcel out those factors which have an effect. And that has been done, for instance, in mice, very nice publication, where they had a large database of a very simple response, and they parceled out all the different factors, genetic, environmental, their interaction, and even in environmental factors which were actually contributing, like handling, lighting conditions, and what have you. And that means basically that it's not so much like you would have to eliminate everything, but rather using that and then having a proper statistical analysis. And thereby, I think you can use to the max what you're interested in. If that is answering the question of.
I think so, because I don't see any replies anymore.
Okay.
I think that's it, unless you see something else, Albert. Just checking. Okay. Well, thank you, everybody. Albert, you're muted, so I'm not sure if you're trying to say anything. Still muted. Albert, I can't hear you.
Yes, I'm. Do you
there
hear
you
me
are.
now? Yes.
Yes, we do.
Something was going wrong. So thank you very much for being with us, and well, we can now all go enjoy the weekend. I think.
Well,
So,
at least on this side.
Yes, that's right. We're of course on the other side of the Atlantic. The day still continues. So thank you very much for joining us all.